Welcome to this lecture on protein databases. Imagine trying to understand the machinery of life without a manual. That is what biology was like before these massive digital libraries existed. Today, we will explore how scientists store, search, and analyze the building blocks of life and the mathematics that make it all possible. Let us start with a simple analogy. Imagine you are a detective trying to identify a suspect using only a single fingerprint found at a crime scene. If you had to knock on every door in the world to find a match, it would take lifetimes. But with a centralized fingerprint database, you can find the match in seconds. In computational biology, scientists face a very similar problem. When they discover a new protein in a cell, they need to know what it does. A protein database acts exactly like that centralized detective agency, saving years of laboratory work. Now that we understand the purpose of these tools, let us define what they actually are. Formally, a protein database is a large, organized digital repository. It stores critical information about proteins, including their linear amino acid sequences, their complex three-dimensional structures, and their biological functions. By comparing a new, unknown protein against this massive library, researchers can instantly guess its function, where it comes from, and how it might behave. This capability forms the absolute foundation of modern biological research, allowing computers to rapidly search, compare, and retrieve biological data using specialized algorithms. Building on that definition, we can categorize these repositories into two main types. First, we have primary databases. These are the foundational archives that store raw, linear amino acid sequences, exactly as they are decoded from genetics. Second, we have secondary databases. These do not just store raw data, they store derived information. This includes conserved evolutionary patterns, structural motifs, or three-dimensional structures built from the primary sequences. Understanding this distinction is crucial because a biologist will choose a different database depending on whether they are looking for a simple sequence match or a complex structural similarity. To make this concrete, let us look at three specific examples of these databases in action. First is SwissProt, which is highly respected because it is manually curated. This means human experts verify every single entry for accuracy, ensuring high-quality data. On the other hand, we have Tremble. Because new biological data floods in faster than humans can review it, Tremble is automatically generated by computers to handle the massive volume. Finally, there is the Protein Data Bank, or PDB. Unlike the others, PDB specifically stores the complex three-dimensional shapes of these molecules, which is vital for understanding how proteins physically interact. Having discussed these specific databases, let us visualize how data flows between them. On the screen, you see a flowchart representing the protein database data flow. On the far left, we start with raw DNA and protein sequences. An arrow points to the right, leading into Tremble, representing the unreviewed, computer-annotated data. From there, another arrow points to SwissProt, showing the transition to manually curated and reviewed data, symbolized by a human expert icon. Below this linear flow, we see a separate box for the protein data bank, containing a complex ribbon diagram of a three-dimensional protein structure. This shows how raw sequences are progressively refined and structurally mapped. Beyond just storing data, this flow of information is absolutely critical for modern medicine, agriculture, and biotechnology. When a new virus emerges, such as the one that caused the recent global pandemic, scientists immediately sequence its proteins and search for them in these databases. If they find a match or a close relative, they can quickly understand how the virus infects human cells and begin designing vaccines or drugs to block it. Without these databases, every single biological discovery would have to start from absolute scratch, costing billions of dollars and decades of time. They also allow us to study evolution by comparing human proteins to those of other animals. Now that we appreciate their biological importance, let us shift gears and look at the mathematics of database searching. When we search a protein database, we are often looking for a specific sequence of amino acids, which are the fundamental building blocks of proteins. Since there are exactly 20 standard amino acids in nature, we can calculate the probability of finding a specific sequence purely by random chance. This mathematical foundation helps us understand if a match we find in a database is biologically meaningful or if it is just a random coincidence. To calculate this random chance, we use a fundamental probability equation. Conceptually, this formula comes from multiplying the independent probabilities of finding any specific amino acid at each position in a chain. In this equation, the symbol p represents the probability of a specific sequence occurring purely by random chance. The number 20 represents the 20 standard amino acids that can occupy any given position. The variable L represents the length of the protein sequence. As the sequence length L increases, the probability P decreases exponentially. This means it becomes incredibly rare to find long matching sequences by pure chance, giving us confidence that long matches are biologically significant. Let us apply this formula with a concrete example. Suppose you are a researcher who has identified a very short protein motif consisting of exactly 3 specific amino acids in a row, such as alanine, glycine, and serine. We want to know the probability of this exact sequence appearing purely by chance. We are given the sequence length, L equals 3. We choose our probability formula and plug 3 into the exponent. This means we multiply 1 over 20 by itself 3 times. The calculation gives us 1 over 8,000, or 0.000125. Physically, this means there is a 1 in 8,000 chance of a random match, which is relatively high, so a 3 amino acid match is usually not considered biologically significant on its own. Because short sequences can occur by chance, knowing the raw probability is not enough. Once we know the probability of a sequence occurring by chance, we need to know how many times we might accidentally find it in a massive database. This brings us to the expected value, or e-value. The e-value is a fundamental concept in database search tools like BLAST. It tells us the number of random matches we should expect to see in a database of a specific size. Understanding this expected value helps scientists filter out the noise of random matches from the signal of true biological relationships. To find this expected number of matches, we use a simple multiplication formula. Conceptually, if you know how rare an event is, and you know how many chances it has to occur, multiplying them gives the expected occurrences. Here, the symbol E stands for the expected number of random matches we will find. The variable N represents the total size of the database, meaning the total number of amino acid letters stored in the system. The variable P is the probability of the sequence occurring by chance. If the database size N increases, the expected number of random matches E also increases, because a larger database gives us more opportunities to find accidental matches. scientists look for a very low e-value, close to 0. Let us see how this works in a large database. Imagine you are searching for a slightly longer sequence of 5 amino acids in a database containing 10 million amino acid residues. We want to calculate how many times we expect to find this sequence purely by random chance. This is a two-step process. First, we must calculate the probability, p, for our 5 amino acid sequence. We use our first formula, plugging in L equals 5. Raising 1 over 20 to the power of 5 gives us 1 over 3.2 million. This tells us that finding this specific 5 amino acid sequence by chance is a much rarer event than our previous 3 amino acid example. Now that we have our probability, we can complete the second step of our calculation. We need to find the expected number of matches, E. We are given the database size N is 10 million, and we just calculated P is 1 over 3.2 million. We substitute these values into our expected value equation. We are essentially asking, if an event happens once every 3.2 million times, how many times will it happen in 10 million tries? Multiplying these values gives us a final expected count of 3.125. Physically, this means you should expect to find this exact sequence about 3 times, purely by accident. Therefore, a match here might just be random noise, and you would need a longer sequence for a reliable result. We have covered a lot of ground today, so let us summarize the most important ideas. First, protein databases are the foundational libraries of computational biology, storing the sequences, structures, and functions of millions of proteins. Second, because there are 20 standard amino acids, we can mathematically calculate the probability of any sequence occurring by random chance. Finally, by combining this probability with the size of the database, we calculate the expected value, or e-value, which allows researchers to confidently separate meaningful biological connections from random noise. You now understand the core principles of how modern biologists query and interpret the molecular machinery of life.